Research into aging and extending lifespan. The mounting evidence for age genes that influence the aging process is by no means conclusive, but it is quite impressive, coming from a variety of independent research, from aging in worms and fruit flies to antioxidants and gene repair mechanisms and human mutations. Still, the connections are circumstantial. Christopher Wills, professor of biology at the University of California in San Diego, thinks that by 2025, science will likely isolate the mammalian age genes in mice. We share roughly 75% of our genes with mice and have much the same body chemistry. This is a strong reason to believe that an age gene found in mice could also be at work in humans. If such genes are located, the next step would be to find out if these age genes have their counterparts in humans. Wills believes that if they are found in humans, they may extend the human lifespan perhaps to 150 years. But by 2020, when personalized DNA sequencing becomes widespread, a second tactic may prove fruitful as well. By analyzing populations of healthy individuals in their 90s and beyond, scientists will find it possible to use computers to compare their genetic backgrounds and cross-check for similarities in key genes that are suspected of influencing aging. A combination of studies on the DNA of long-lived animals and on the personalized DNA sequences of elderly individuals may considerably narrow down the search for the age gene. As yet, none of these methods can prove that we can increase the human lifespan. Indeed, the only theory with a proven track record of extending the lifespan of animals is the caloric restriction theory, which states that animals which consume calories just above starvation levels live significantly longer than the average. Although this theory flies in the face of common sense, a well-fed animal is well-nourished and healthy and should have greater resistance to disease and aging. It has held up under repeated testing among a wide range of animals. Scientists have consistently increased the lifespan of rats and mice in the laboratory by 50 to 100%. It is the only laboratory-tested theory of age extension for animals that has held up under decades of careful scrutiny. Across the animal kingdom, the lifespan of animals is roughly inversely correlated to the metabolism rate. The slower their normal metabolism rate, the longer their normal lifespan. In 1996, in a study that reduced the calorie intake of 200 monkeys by 30%, the monkeys were shown to have a slower metabolism rate, a longer lifespan, and reduced rates of cancer, heart disease, and diabetes. We have known for 70 years that if you feed laboratory mice less food, they age slower, they live longer, and they get diseases less frequently. We find that monkeys respond in the same way as rodents, and that the same biological changes may be in play here, says George Roth of the National Institute of Aging. There is still room for scientific debate on the question, why? Ron Hart, a scientist at the National Center for Toxicological Research, believes that the answer may lie in the high body temperature of mammals, and humans in particular. Heat causes pieces of the DNA molecule to split off randomly, and it must be repaired, Hart says. Under calorie restriction, though, the engine runs cooler and there's less damage. Merely reducing caloric intake by 40% reduced this form of spontaneous DNA damage almost 24%. Furthermore, at a higher internal body temperature, oxygen is being burned at a greater rate, creating more free radicals, which also speed up the aging process. Cooling the body, on the other hand, increases the amount of antioxidants in the body. Hart found a 4-fold increase in the enzyme catalase and a 3-fold increase in superoxide dismutase in animals on a restricted diet. What's fascinating, Hart concludes, is that reduced food intake is the only experimental paradigm ever found that enhances DNA repair. Hart is so convinced of the importance of this work that in 1993 he began the first systematic studies of caloric restrictions in humans.